Would you rather me use a microphone like this? Okay. I was going to try the option without it, but I'm not. All right, is this good? Can everybody hear me okay? Yes? All right, so welcome. This is a final exam review for Biology 1. So hopefully we're all in the right place. I'm just going to be going through a practice exam. If you don't already have it, you could find it on this website here. All you need to do is go to the biology section and you should see Shannon somewhere, and then I have a link to it underneath it. So don't worry about trying to write all the questions down. Everything's online there for you. So all right. So everybody got the link? We're good? I'll give you a second. Are we good? I'll give you one more second. If you don't have the link, take a picture. All right, let's get started. All right, number 1. Which of the following are in the correct order? Right away I can see that it's A. I like to use an acronym to remember which order is the correct order. So usually, remember this by saying King Philip came over for good soup. So that stands for kingdom, phylum, class, order, family, genus, species. So all of the rest are in the wrong order. Number 2, which of the following is true? I think this one is fairly obvious, but D. Protons are positive. Just in case you need to review, electrons are negative, neutrons are neutral, and protons are positive. 3, an isotope differs in the amount of which of the following? That would be neutrons. If we differed in the number of protons, that would make it a different element. And if we differed in the number of electrons, that would make it an ion of some sort. Number 4, oxygen is number 8 in the periodic table. How many valence electrons does it have? So if oxygen is number 8, that tells us it has 8 electrons. So if we want to figure out the valence electrons, we would have to draw the orbitals. So the first orbital is going to hold a maximum of 2. So I'm going to draw my 2 electrons in. I'm going to have another row. And since we have 8 total, I'm drawing the wrong box. If we have 8 total, that means I'm going to have 6 more. 6. Giving us a total of 8. So how many valence electrons do we have? It would be 6. And sorry, I drew it in the wrong space. But you get the point. Just make sure you don't take any guesses and you draw the orbitals for this one. Number 5. What is the maximum number of electrons orbitals 2 and beyond can hold? We kind of talked about that a little bit with the previous one, but that would be 8. First orbital can hold a maximum of 2. Everything after that can hold 8 electrons max in the orbitals. Number 6, a bond where electrons are shared. That would be a covalent bond. In ionic bonds, electrons are transferred. Electrons are transferred in ionic bond, and hydrogen bonds are formed from a result of polar covalent bonds. And obviously it's not James Bond. Number 7. Cohesion is between water molecules. What would B be? Adhesion. Alright, number 8. If a pH of a liquid is 8, what is the hydroxide concentration? So they told us pH equals 8. So the first thing I'm going to do is convert that to a pOH. So my pOH would be 6 because I'm going to subtract that from 14. Because we know that pH and pOH always add up to 14. And since it asks us for hydroxide concentration, that means we need to go one more step further and convert that to a scientific notation number. So if we're finding hydroxide concentration, it's going to correspond with the pOH. So it's going to be 1 times 10 to the negative 6, which is D. So remember that pH and pOH always add up to 14. Alright, number 9. This time they tell us the hydrogen ion concentration is 1 times 10 to the negative 5th, and we want to find the pOH. So they told us the H plus concentration is 1 times 10 to the negative 5th, and we want to find the pOH. So the first thing I'm going to do is find the corresponding pH that goes with this hydrogen ion concentration. So we see that there's a 5 as the exponent, so that means our pH is going to be 5. And then if we want to find pOH, we're going to subtract that from 14 like we did in the last problem. That way we know that our pOH is 9, which is B. So all you need to know for pH problems is that pH and pOH are always going to add up to 14. They're always going to be positive whole numbers. And if you're finding a concentration, we know concentrations are always those small numbers in scientific notation. Alright, number 10. What is a structural isomer? That would be A molecule that differs in connectivity of atoms. If a molecule differed in spatial arrangement, what would that be? An enantiomer. And what about if it was a molecule that differed in double bond arrangement? What would that be? Geometric or cis-trans? Or cis-trans. Yes. Bigger? Is that better? Alright, 11. What functional group is this? This, we know, is a carboxyl. We know it is a carboxyl because we see that it has a double bonded O to a C, and directly connected to that C double bonded O is an OH. If this were a carbonyl, it would just look like this. There would be no OH. So this right here tells us it is a carboxyl. Think of a carboxyl as a carbonyl plus a hydroxyl. because by itself the OH would just be called a hydroxyl. Alright, 12. A condensation reaction builds molecules. What is it called when we break down molecules? Hydrolysis. So condensation builds molecules, hydrolysis breaks down molecules. Another name for condensation would be dehydration. So I'll write that down. So dehydration synthesis is another name for condensation reaction. What type of bond connects two sugars and a carbohydrate? What does an ester linkage connect? Lipids. What does a peptide bond connect? Proteins or amino acids. What does a glycosidic linkage connect? Carbohydrates, so that would be our answer. And then hydrogen bond is a hydrogen bond. So yes, a glycosidic linkage is what connects two sugars in a carbohydrate. Which of the following is not a type of lipid? It would be C, an enzyme, because an enzyme is a type of what? Protein. What three things do all amino acids have? Do all amino acids have an R group? Do all amino acids have a hydrogen bond? No. So it's not A. Do all amino acids have a carboxyl group? Yes, they do. Do all amino acids have an amino group? Yep. Do all amino acids have an R group? Yep, so B is our answer. Which level of protein structure do alpha helices and beta plated sheets form? This is going to be secondary. What type of bonds do we use when we're making beta plated sheets and alpha helices? Hydrogen bonds. What type of bonds do we use in primary structure? Peptide bonds. In tertiary, we're going to have lots of different types of bond. You could find any of them there. And then quaternary is optional. I think they like to emphasize, though, that there are a lot of covalent bonds formed in tertiary. But you can find others there, too. What differs between RNA and DNA? The sugar. I have no idea what color they are. And we know that since they're both nucleic acids, they're both going to have a phosphate group. They both have a phosphate group and a sugar, but their sugar is going to differ. So what sugar does RNA have? Ribose. What sugar does DNA have? Deoxyribose. 18. Which organelle is responsible for detoxifying substances in the cell? That is going to be peroxisomes. Peroxisomes detoxify. Remember the ox. Peroxisome, detoxify. What do lysosomes do? Degrade. So it's very easy to confuse the two. So remember, detoxifies peroxisomes. Lysosomes degrade or digest. And why not? What do mitochondria do? Cell respiration, so they make ATP. Remember, they're the powerhouse of the cell. And the rough ER does what? Right. They have ribosomes, so they make proteins. Which of the following is present in animal cells but not plants? So this one is going to be centrioles. Both plants and animal cells are going to have rough ER, mitochondria, and cell membranes, but only animal cells are going to have centrioles. However, both plants and animals have centrosomes, but only animal cells have centrioles within their centrosomes. So centrosomes are both plants and animals, but only animal cells have centrioles. Number 20. Which of the following cytoskeleton proteins is the smallest? These will be our microfilaments. Which ones are our largest? Microtubules. So then obviously that makes intermediate filaments the intermediate ones. They're in the middle. Which of the following needs ATP? Active transport. Think it's active, it needs energy. If it's passive, it does not need energy, it's just passive. It does what it wants to do. And osmosis is a type of passive diffusion, although specifically dealing with water. All right, 22. A cell is in a solution. The solution is 20% salt. The cell is 30% water. Is the cell hypotonic, isotonic, or hypertonic to the environment? So we want to find the cell relative to the environment. So if they told us our cell is 30% water, that means it is also 70% salt. Similarly, if the environment is 20% salt, that makes it 80% water. So our cell has a lot more percentage of salt than the environment. So if our cell has a higher concentration of salt, that makes it hypertonic. If we wanted to, we could compare the waters also. We see the cell has 30% water, but the environment has 80%. If the cell has less water than the environment, that also makes it hypertonic. So if something has more salt or solute concentration, that makes it hypertonic. If something has more water, that makes it hypotonic. So if we wanted to, we could look at the environment and see we could see the environment has more water than the cell. So if that means that the environment has more water, that means the environment is hypotonic. If the environment is hypotonic, that means the cell is hypertonic. If something is hypertonic, it will swell because that means water will rush into the cell. Question? So if we want to state where water would move after we figured this out, We know that water would go in because there is less water there than the outside. So water's going to move from outside in. Water always moves towards the hypertonic area. So if the cell is hypertonic, it's going to swell. So this is 22. 23. A cell is in solution. The solution is 60% salt. The cell is 40% water. Is the cell hypo, hyper, or isotonic? So if the cell is 40% water, we know that the cell is also 60% salt. If the environment is 60% salt, we know that it is also 40% water. Both inside and outside of the cell, we have 40% water and 60% salt, so that makes this isotonic. If both the concentrations inside the cell and outside the cell are equal, they're going to be isotonic. Iso meaning same. I'll give you guys a second to write that. All right, 24. A catabolic pathway is exergonic. Endergonic, a pathway that builds molecules, and a positive delta G all correspond with what? Anabolic. So these are all anabolic properties. So if we wanted to say what to find a catabolic pathway besides being exergonic, we would say it's going to have a pathway that breaks molecules, and it's going to have a negative delta G. So pretty much the opposite. (25) Which step of cellular respiration makes the most ATP? The electron transport chain. Roughly, how much ATP does the ETC make? 32, 34, something like that. And how much do glycolysis and the Krebs cycle both make? 2. So obviously, the electron transport chain definitely makes the most. It makes way more than the other two. (26) What molecule combines with oxaloacetate to begin the citric acid and the Krebs cycle? This is going to be our acetyl-CoA. So you might be tempted to pick pyruvate, but remember, before the Krebs cycle started, we converted our 2-pyruvate to 2-acetyl-CoA. And you might also get a little thrown off by the citrate because remember, our acetyl-CoA combines with oxaloacetate, and those yield citrate. And that's where the citric acid cycle gets its name from. (27) What is the name of the protein involved in the electron transport chain that produces ATP? ATP synthase. (28) Does NADH or FADH2 enter the electron transport chain first? NADH. We make more of NADH than FADH in the Krebs cycle because it's better. It's going to enter the electron transport chain first, and it's going to have a greater yield of ATP. #29. Where does glycolysis take place? Glycolysis takes place in the cytoplasm. I don't know if you guys need to know 30, but we'll go through it just in case. Because it has to do with photosynthesis, you might not have covered this. But which enzyme combines carbon dioxide and RUBP to begin the Calvin cycle? That is rubisco. Did you guys talk about that at all for photosynthesis? Don't worry about it then. Ignore number 30 if you have Professor Thomas, because I know she doesn't talk about that. Alright, 31. If an animal's diploid number is 16, how many sister chromatids will be present in anaphase of mitosis? So if we have 16 chromosomes, that means when we are replicated, we'll have 32 chromatids. In anaphase, we are technically unreplicated, but since everything's in the same cell, we're going to have 32 chromatids. So, if this is metaphase, all of our chromosomes are going to be lined up. And I'm not going to draw all 32 chromatids, but you can imagine that they're there. And if we're then in anaphase, we're going to have pulled those apart. But notice we still have the same number of chromatids. So if we know that there were 32 chromatids in metaphase, since we're replicated, then we know we will also have 32 chromatids in anaphase. So if you're ever stumped with these kind of questions on the test, draw a picture with like a couple of chromosomes and compare that to what your number would be. #32. If an animal's diploid number is 4, how many homologous pairs will be present in prophase 1 of meiosis? So if we have 4 chromosomes, that means we're going to have 4 X's. And a homologous pair is made up of 2 replicated chromosomes, or 2 X's. So this would be 1, and this would be 2. So if we have 4 chromosomes, we know that we have 2 homologous pairs. So this is a homologous pair. #33. If an animal's diploid number is 6, how many chromosomes will be present in telophase 1 of meiosis? So, in telophase 1, we're not yet haploid because our cells have not been cut in half. And we're just asked how many chromosomes. So, we're going to have 6. We're going to have 6 because nothing has really happened by telophase 1. In the metaphase 1, we're going to have all 6 of our chromosomes lined up. Ooh, scratch that. We're going to have homologous pairs lined up. So this would be metaphase 1, because we're talking about meiosis. So metaphase 1, our 3 homologous pairs, since we have 6 chromosomes, are going to be lined up. And when we go through anaphase 1, our homologous pairs will be pulled apart. But if we're still in the beginning of telophase 1, we'll still have 6 chromosomes because that cell has not yet been cut in half. If this question had said after telophase 1 and cytokinesis, then we'd only have 3, because this would have been cut in half. But we're saying in telophase 1, so it has not yet completed. So make sure we understand that we're in meiosis, so that we start off with homologous pairs. And we know in anaphase 1, we pull apart homologous pairs, and we separate those into 2 different cells by the end of meiosis 1. Okay? So we'd still have 6 chromosomes because telophase 1 is not yet completed. All right, 34. Germ cells is another word for sex cells. What would the other 3? Are the other 3 all the same thing? That's a bad question. But yeah. Body cells and skin cells are both examples of somatic cells. When does crossing over occur? So the page printed at a terrible spot. We have to locate D over here, because it's at prophase 1. So crossing over always occurs at prophase 1 of meiosis 1. All right, 36. A plant with genotype big A little a is crossed with a plant with a genotype big A big A. We want to find the percent chance of getting an offspring that is homozygous recessive or little a little a. So all I'm going to do is put these in a Punnett square and do my cross. So the percent chance of getting homozygous recessive would be 0. There's no possible combination that somebody could be homozygous recessive unless both parents had at least 1 copy of the little allele. So if you wanted to save yourself a little bit of time on the exam, you could see, oh, we don't have a little allele in both parents, so we can't possibly make this. I mean, it doesn't take a whole lot of time just to make the Punnett square, but if you wanted to save a little bit more time, that could be a way of doing it. So that is 36. Draw the Punnett square for the cross between big A, big B, and big A, little b. And then we'll go back to the questions after we draw. So the first thing we need to do is, well, we have to realize this is a dihybrid cross. We're dealing with 2 different traits. We have trait A and trait B. So what we're going to have to do is we're going to have to figure out what possible gametes or what possible combinations both individual can make. This one can make big A, big B, or little a, big B. The way we find that is by foiling. If we foil this out completely, we would see that the only 2 different things we could make are these here. Similarly, the other individual can only make big A, little b. There's no other possible combinations this person could make. If we want to verify that we've made the correct number of different combinations, we could use the 2 to the n rule. So the 2 to the n rule is N is going to be the number of heterozygous combinations that individual has. So for the first one, they only have one. So we do 2 to the power 1, which is 2. So it tells us that individual can make 2 different possible combinations, which we have. The other one doesn't have any. So we would do 2 to the 0, which is 1, which tells us that individual makes a possibility of 1 combination. And we got it. So you don't have to use the 2 to the n rule, but you can, just to make sure that you've got all the information that you need to do your Punnett square properly. So now that we've figured out our possible combinations, we can draw our Punnett square. So these would be the 2 possible offspring we could make. And our question says, what % of the offspring will be big A, big B, little b? In this case, we see that we have that one right here. So it's going to be 50%, 1 out of the 2. They also ask us what % would be heterozygous for both. We see that we have 1. So there's going to be a 50% chance of being big A, big B, little b. Okay? I'll give you guys a second to write that down. Everybody got this? I'm going to move it. I'll give it another second. Good? Alright, pedigrees. Number 38. Label the genotype for everyone, assuming the trait in black is a recessive disease. Ignore individuals 4 and 5 in generation 2. So we're going to ignore these because that doesn't make sense. So we know that if they're shaded in, they have a recessive disease. And if we have the recessive disease, that means we have to be homozygous recessive. So I'm just going to use A, and that means we automatically know anybody shaded in is going to be homozygous recessive. Because in order to have the recessive trait, you need to have both, 2 small alleles. So if we want to figure out the others, we're going to have to do a little bit of figuring out. So in generation 1, if these 2 individuals can produce an offspring that has the trait, what does that tell us about this man here? He has to be heterozygous. In order for him to have an offspring that has both little alleles, would mean that he had to have passed one on to his son who has the disease. And we know that he has to have at least 1 big A because his box is not shaded in. So he does not have the disease. So from that, we know that he would be heterozygous. We could also figure out this female offspring. We can figure out her genotype because we know that mom could only pass a little a. And if she's not shaded in, that means her other allele is going to have to be a big A. So we figured her out also. I'm going to go with the assumption that anybody marrying in, or wait, never mind, we have enough information to figure this one out. Yes. So if we didn't have enough information, we would assume somebody marrying in would be genotypically normal. But in this case, they give us enough information to figure out what this person marrying in's genotype is going to be. So once again, if these 2 people can have an offspring that's shaded in that has the trait, what does that tell us about this female who's marrying in? She's going to have to be heterozygous. Because in order to have an offspring who's shaded in who has the trait, both mom and dad would have had to pass a little a. So we were able to figure out her genotype in this case. So that also tells us that this female offspring is going to be heterozygous because dad passed her a little a, because we know that's the only thing he can pass. And since she's not shaded in, she's going to have at least one big A. And then we can do the same thing to figure out this female marrying in. If they have offspring that are shaded in, she's going to be heterozygous just like the others. And if she is heterozygous and the husband is homozygous recessive, that means their offspring will either be a carrier or they'll have it. And obviously this stuff we shaded out here couldn't work, because if these two people were both homozygous recessive, they couldn't possibly have offspring that don't have the trait. So obviously we can't figure this out because there's some sort of mutation going on here. And we would need more information to figure that out. So that's why we're skipping that. Okay. Alright, 39. If a DNA strand contains 30% thymine, what percentage of guanine does it have? So if it has 30% T, we automatically know it also has 30% A. Because A and T bond together, we know that there's always going to be equal amounts of each. So that gives us a total of 60%. That means we're going to have 40% left over for both C and G. So we're going to split that into equal parts, and then we'll know that there's 20% C and 20% G. Number 40, DNA is anti-parallel. This means that A, the 5' and 3' ends of each strand are going to face each other. If we had 2 5s on one side and 2 3s on the other side, that would just be regular old parallel. But they're anti-parallel because the 5 and the 3 are going to be matching. 41. DNA polymerase 3 adds in which direction? Always 5 to 3. New DNA is always made from 5' to 3'. Never do we make it the other way. 42. Which enzyme unzips the DNA? Helicase. What does topoisomerase do? Prevents overwinding. What does ligase do? Seals the Okazaki fragment. And what does primase do? Places the RNA primer. And I know this section is especially important for you guys because you have a test on this tomorrow. Yay. (43) Which enzyme adds nucleotides? And I should have corrected that, but DNA nucleotides specifically. So which enzyme adds DNA nucleotides is going to be DNA polymerase 3. Yes. Doesn't what? Which one? Yeah, okay. Yeah, you could say that too. Yeah, DNA polymerase 1 would also add nucleotides because it replaces the RNA primer and puts in DNA nucleotides. So yeah, you're right. My bad. What kind of nucleotides does primase add? RNA. (44) Which enzyme makes the mRNA transcript? This is going to be RNA polymerase. So think, what are we making in transcription? We're making mRNA. So it would make sense that the name of our enzyme would be RNA polymerase, not DNA polymerase. We're going to use all of our DNA polymerases in DNA replication. So RNA polymerase is going to go with making mRNA, transcription. (45) Which of the following do not need to occur to make a mature mRNA? This is going to be exon splicing. We don't want to splice out our exons, we want to splice out our introns. So, to turn our pre-mRNA into mature mRNA, we're going to splice out introns, and we're going to add the poly A tail, and we're going to add the 5' cap. Exons are what is going to stay. (46) What is the first site the first tRNA molecule enters in the ribosome? The P site. Remember, the very first tRNA doesn't need to wait. There's nobody in its way. So it gets to go right to the P site. If it were any other tRNA, it would have to wait in the A site, because there will already be a tRNA in the P site occupying it. Alright, 47. Let's say I didn't specify, but I'll tell you it is the non-template. So if our non-template is ATC GGA, then the first thing we're going to have to do to get to our anticodons is to find the template. So our template is just going to be the complementary. It'll be TAG CCT. But we still have some more things to do because we need to get to the anticodons. The anticodons will be the tRNA. So the next thing we're going to do is find the mRNA. Because we know the mRNA is going to be complementary to the template strand. Because that's what RNA polymerase uses to make our mRNA in transcription. So we're going to find the complementary to the template strand, but keep in mind, since we're in RNA, we're going to use U's and not T's. So it'll be AUC GGA. Now that we have our mRNA, we can do our last step and find our tRNA. Since we know our tRNA is the same thing as the anticodons, and we know the mRNA is the same thing as the codons, we know that mRNA and tRNA are going to be complementary. So it'll be AUAG CCU. So these will be our anticodons. I would hope they would specify. I just forgot to write it in. Yeah. 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 I could write that this would be, and then these would all be the opposite. So yeah, keep in mind whenever you make a complementary strand, it's going to be anti-parallel. What do you mean by that? Oh, it'll be the template, yeah. The template will be the coding strand. Because the template is what we make our complementary mRNA off of. Everyone got this? Can I move it? We're good? Alright, 48. What happens at the P side of the ribosome? That's where we attach our amino acid. Think of P for polypeptide or P for protein. That's the site we're making our protein. So I'm not 100% sure if you guys talked about the bacteria and viruses chapter too much. I will give you the answer just in case, so we won't spend a lot of time on them. Okay, so 49. If the infected cell starts producing viruses, that means it's entered the lytic. I don't know if you guys talked about prions. Maybe? Yes, infectious proteins. Awesome. 51. Which of the following is only found in animal cells? Okay, this is something we can do. Did we already have this question? No, we didn't. Was it exactly the same? I think we had something slightly different. Okay. So you kind of could figure this one out based on the last one we did. So which of the following is only in animal cells? This is going to be lysosomes. So remember in the previous question we said that centrioles were only in animal cells, but they both had centrosomes. So don't be tempted by D. Peroxisomes are going to be in both, and cell walls are only in plants. So these are plant specific. And peroxisomes and centrosomes are both. 52. Osmosis is the movement of water. Facilitated diffusion requires the help of a transport protein. I think it's facilitated because that transport protein is helping the diffusion happen. It's facilitating it. Chloroplasts and ribosomes, they're organelles, so they don't really have anything to do with this. The only other answer that might be confusing would be glycolipids. But glycolipids are going to be more for cell recognition, and those like the little things that stick off the outside of the cell membrane. Alright, for 54 through 62, we are going to find the chromatid and chromosome number for each. So I'm going to write the chromatids on the left and the chromosomes on the right, just so we're on the same page. Okay, and our diploid number is 20. So if we're at prophase, we know we're going to have 20 chromosomes. Oh, sorry. I'm doing chromatids on the left, aren't I? Scratch that. We know that we're going to have 20 chromosomes. And if we're in prophase, we're going to be replicated. So we're going to have 40 chromatids. So, chromosomes. If we're in metaphase, it's going to be the same. We're going to have 40 chromatids and 20 chromosomes. If we're at anaphase, we're still going to have 40 chromatids. But remember, at anaphase, we're pulling apart our replicated chromosomes and we're becoming unreplicated. So all 20 X's we had turn into 40 unreplicated chromosomes. So we're going to have 40 chromosomes here. So in prophase and metaphase, we had 20 of these. We had 20 X's. And we know that there's 2 chromatids per X. So that's how we figured out if we have 20 X's, we have 40 chromatids. In anaphase, since we pulled them apart, we have 40 of those. So that means we're going to have 40 chromosomes and 40 chromatids. Because now that our chromosomes are unreplicated, They're only made of one chromatid each. So G1. G1, we know we're going to have 20 chromosomes, but we're not yet replicated in G1. So there's only going to be 20 chromatids. If we're in G2, we're past S phase, so our 20 chromosomes are going to be replicated, meaning we're going to have 40 chromatids. Anytime we've passed S phase and we're before anaphase, we're going to be replicated. Because remember what's going on in S phase. S phase, we're doubling all of our DNA. We're turning our unreplicated chromosomes into replicated chromosomes. So if we had 20 chromosomes, that gives us 40 chromatids. Alright, now in meiosis. If we're in prophase 1, we're going to have 20 chromosomes, and pretty much the same as the last one. We know we're replicated in prophase 1. So we're going to have 40 chromatids. If we're in metaphase 1, it's going to be exactly the same. 20 chromosomes, 40 chromatids. Now if we're in prophase 2, we know that after meiosis 1 we became haploid. But we also know that we're still replicated. So if we're haploid, that means we now have 10 chromosomes, because we cut 20 into 2 different cells. But if we're replicated, we know that we're going to have 10 X's, giving us a total of 20 chromatids. And metaphase 2 is going to be exactly the same. We're going to have 10 chromosomes and 20 chromatids. If I were to ask you at the very end, after telophase 2 and cytokinesis happened, how many chromosomes and chromatids will we have? 10 and 10. Because our end goal of meiosis 2 is to become unreplicated. So, end goal of meiosis 1, become haploid. End goal of meiosis 2, become unreplicated. So that we eventually end up with 4 daughter cells that have half the number of chromosomes we started with and are unreplicated. All right. Everybody got these numbers? You good? Bless you. Alright, 63, Munchkin cats. So Munchkin cats is a dominant condition, but homozygous dominant offspring are not viable, so they'll die in the womb. So if we have 2 Munchkin cats, we automatically know they're going to have to be heterozygous, because Munchkin cat is a dominant trait, so they have to have a big M. But they can't be homozygous dominant because that's not viable. We can't have one of those. It doesn't exist. So if we have 2 Munchkin cats breeding, we know they're both going to be heterozygous. So once we figured out their genotypes, we can do a Punnett square. And before we even look at the question, we are going to cross out anything that's homozygous dominant. If something is not viable, we're not going to use that in our percentages. If it can't possibly be produced, then it's not even an option. So we're going to cross out our homozygous dominant in this case because we know it's not viable. So then they ask us what percent will be genetically normal. In this case, genetically normal means homozygous recessive, because the trait we're talking about is the dominant trait. So genetically normal means we don't have the dominant trait. That means we have to be homozygous recessive. So we see that we have 1, but we're going to make sure that we say it's 1 out of 3 and not 1 out of 4, because we crossed out homozygous dominant. So you can say we have a 1/3 chance or 33% chance of being a genetically normal cat. So really the question is pretty simple. We just have to make sure that you know if something is not viable, we do not include it in our percentages. Alright, 64. 64 says, in calico cats, why is it that only the females show the calico pattern? And in what situation could a male be calico? So, we have to know that calico cats are X-linked. So if we have a calico cat, our female cat is going to have one X carrying the orange trait and one X carrying the black trait. If we had a male, he could only have one or the other. So we have to understand that since the Y cannot carry a trait, that male cat can't be calico. Because he'll only ever have the black or the orange trait because his X is the one that carries the trait. So that's why only females carry it. So if we want to know what situation a male could be calico, we have to think a little further and know that it's going to be some kind of mutation. So if a male cat were to be calico, he would have to be XXY and have an extra X in order to display the calico pattern. Anybody know what it's called when you have an extra X chromosome in a male? Klinefelters. So the only situation we could have a male calico cat is if he had a mutation that caused him to have Klinefelters. Normally, a male cat could only be orange or black. Everybody have this? Can I take it away? We good? '65. Can men or women be hemizygous for a trait? Men. To be hemizygous means that you're only carrying an allele for one trait. Or you're only carrying one allele. Because the Y doesn't carry anything. So whatever that X carries is going to be what that male displays if it's X-linked. So that's what it means to be hemizygous. It means we only have one allele because the Y does not carry anything. All right. '66. A colorblind male produces offspring with a heterozygous female. What are the chances they have a daughter that is colorblind, and then what about a male that is colorblind? So if the male is colorblind, we have to know one thing, and that this is also an X-linked trait. So if the male is colorblind, we know that his X is going to have the recessive allele. Although they didn't tell you that. But we also have to know not only is colorblindness X-linked, but it's also recessive, which I should have wrote here, but I did not. So if our male is colorblind, we know his X is going to carry the recessive allele. And then they tell us the female is heterozygous. So one of her X's is going to have the big allele, and her other X is going to have the little allele, which is going to be heterozygous. And we want to figure out the chances they have a daughter that is colorblind or a male that is colorblind. So the first thing we're going to do, now that we have their genotypes, is do a Punnett square. And do the cross. So first of all, we want to find out the chance that they have a daughter that is colorblind. So if we want to figure out, I can't hear you. Oh, yeah. So if we're trying to figure out just the females, we're just gonna exclude the males. And out of the females, we're gonna see how many are colorblind. So out of the females, we have 1 out of the 2 being colorblind. So that means there is a 50% chance of a colorblind female. And we're going to do the same thing when it asks us about a male that is colorblind. We're going to exclude the females and only look at the males. And we see that out of the males, one of the 2 is colorblind. So similarly, we have a 50% chance of a colorblind male. So make sure that when you're doing an X-linked problem and they ask you for a specific gender, we're going to exclude the one we don't want and only look at the gender they asked us and find our percentages from there. If this was a autosomal problem, we don't know anybody's gender by looking at their genotype, so we would just take the overall chance of having the trait and multiply that by 1/2. But since we know the gender right from the genotype, we can just look at it from there. Do you have a question? Right. Yes. Yeah, we only multiply by 1/2 if it's autosomal. Okay, 67. Woman has blood type A, and a man has blood type B. They produce an offspring with O type blood. How is this possible? The only way we'd be able to do that is if the woman was AO and the Dow was BO. Because remember, when we're talking about blood type, A and B are co-dominant, so they're both dominant over O, which is recessive. So if we want somebody with O type, and we want to prove that this will make an O blood type offspring, we could do the Punnett square. And we see that we can indeed make somebody with O type blood in this this group, this combination. That's the word I was looking for. So both mom and dad would have to carry the O allele to have an offspring with O type blood. Because in order to have O type blood, we have to have 2 O alleles because it's recessive to both A and B. Everybody got this written down? We good? All right, 68. A nonsense mutation causes what? A premature stop codon. What would B be? If it's not a nonsense, what is it? A missense. What would C and D both cause? A frame shift. A simple base substitution would cause what? Could it cause a premature stop codon? Yeah, it could. Could it change to the wrong amino acid? Yep. Could it cause an error in the reading frame? No, it could not. The only time we get an error in the reading frame is if we had an insertion or a deletion. If we just have a simple substitution, we're not messing up our reading frame because we still have the same number of nucleotides. So that tells us it's not A and C, so D is going to be our right answer, A and B. 70. Which of the following take place in the cytoplasm? Does transcription take place in the cytoplasm? Where does it take place? In the nucleus. Does translation take place in the cytoplasm? Yes, it does. Does the electron transport chain take place in the cytoplasm? Does the citric acid cycle take place in the cytoplasm? Where do both C and D take place in? The mitochondria. So that means B is our answer. Right. Yes. If we were talking about prokaryotes, everything would have to happen in the cytoplasm because prokaryotes do not have a nucleus. So keep that in mind. Although for you guys, the transcription that you learn is, I believe, eukaryotic because we talk about it being in the nucleus. I'm assuming translation is also, but I know for DNA replication you learn prokaryotic replication. What organelle is responsible for producing proteins? Ribosomes. Ribosomes make proteins, and we know that a lot better now because we've talked about translation recently. We've talked a lot about those ribosomes making proteins. 72. Which organelle organizes the microtubules in the cell and plays a major role in mitosis and meiosis? Once again, it's always a terrible place for the page to cut off, but that is going to be centrosomes. We'll go back to you guys might not have gone over 73 and 74, so I'll just tell you the answers. So viroids only infect plants, and conjugation is when genetic material is passed directly from one bacterium to another. 75. Water has a high specific heat. This means that B, it fluctuates very slowly compared to other liquids. So think about the ocean in wintertime. The ocean in wintertime is usually much warmer than the air when it's really cold at the beginning of winter. '76. Which level of protein folding does not occur in all proteins? We kind of talked about it from a previous question, but it would be D, quaternary. Quaternary is when we have multiple proteins that have gone through primary, secondary, and tertiary combining. So I know the example they like to use in your textbook is hemoglobin. Hemoglobin has gone through quaternary structure because it is made up of 4 different polypeptides attached. So quaternary does not always happen. Okay, 77. Unsaturated fats are they solid at room temperature? No. Are they liquid at room temperature? Yes. Do they have a double bond? Yes. So that means D is wrong. So 77 would be B and C, which is choice F. Unsaturated fats are liquid at room temperature and they have a double bond. Saturated would be A and D. '78. Peripheral proteins are B, bound to the surface of the membrane. So we know they're peripheral because they're on the periphery. They're on the outside. If they were located embedded in the cell membrane, that would be an integral protein. So A is an integral protein because it is integrated in the membrane. Receptor-mediated endocytosis is, well, first of all, we know endocytosis is bringing something into the cell. And if it's receptor-mediated, that means it needs a receptor. So that means C is going to be our answer. Receptor-mediated endocytosis uses receptors to bring in specific molecules. What is A, bringing in food? What would that be called? This is phagocytosis. B would be penocytosis, bringing in liquids. And no, it is not a term for exocytosis. Exocytosis is something leaving the cell. All right, number 80. A reaction is spontaneous when B, it has a negative delta G. Positive delta G, endothermic and anabolic would all correspond to being non-spontaneous. So remember, if something is endothermic and has a positive delta G, it needs energy to happen. So something that releases energy is going to be more spontaneous of a reaction. It's more likely for something to release energy or break than it would be for something to need energy and just happen. So that's why spontaneous corresponds to having a negative delta G or releasing energy. #81. If a molecule binds to an enzyme at a site that is not the active site, it is an example of non-competitive inhibition. If it was competitive inhibition, the competitor would be binding at the active site. It'd be directly binding to the active site. That would be competitive. But since it's binding somewhere that's not the competitive site, it is non-competitive. Or sorry, not the active site, it is non-competitive. #82. If an element becomes more negative, it is being reduced. Think reduced. It's being brought down. It's being reduced. It's becoming more negative. #83. During cellular respiration, NAD plus becomes reduced because it goes from being NAD plus to NADH. It went from being positive to neutral. Therefore, it became more negative or less positive, which in this case is reduced. #84. Where does the Krebs cycle take place? The mitochondrial matrix. What happens in the cristae? The ETC. And then the cytoplasm would be what? Glycolysis. And the stroma that's in a chloroplast, so it does not apply here. #85. Which checkpoint during interphase is often called the restriction point? This is going to be G1. The restriction point is important because after G1, we're going to go through S phase. In S phase, we replicate all of our chromosomes. So we want to make sure we're absolutely ready to move on to the S phase after G1, because once we get to S phase, we're pretty much committed to doing the rest of the cell cycle. If the cell decides it doesn't want to go through mitosis or meiosis, it's a lot easier to stop at the end of G1 than it would be somewhere else. So that's why we say that is the restriction point. '86. Cancer cells are density independent. Cancer cells do not care how many other cells are around. They will continuously divide regardless. Whereas a normal cell, if there wasn't room for it to divide, it would stop. Similarly, cancer cells would also be anchorage independent, meaning they don't need to hold on to something to divide. Whereas normal cells would need to anchor themselves somewhere in order to go through cell division. So cancer cells break the rules, and they could do it their own way. So cancer cells are density independent, anchorage independent, and division unlimited. So cancer cells can divide unlimited, no end. '87. If a red flower and a white flower produce offspring, and it is known that they have incomplete dominance, then the offspring will look pink. When we have incomplete dominance, that means one trait is not completely dominant over the other, so we're going to have a mixing. So pink and white mixed together, or sorry, red and white mixed together would make pink. If it was co-dominant, which of these answer choices would be picked? A. Co-dominant means both are dominant, so we're going to see both. So if it was co-dominant, we'd see patches of red and patches of white. So this right here is co-dominant. I can't spell dominant. '88. Blood type is an example of what? Co-dominance. We know that A and B type blood do not dominate over each other. So we could have type A, we could have type B, and we could have type AB, because if we have an A and a B allele, we're going to display both of them. They are co-dominant. '89. Which pair of chromosomes are not homologous? The X and the Y. And in humans, that would be pair 23. Because we know that Y chromosome is a little bit smaller, and it's not going to be the same as the X chromosome. Number 90. Are sister chromatids always identical? Yes, they are. Sister chromatids are always identical. But number 91, are homologous chromosomes always identical? No. So yes, homologous chromosomes are homologous to each other, meaning they both carry genes for all the same traits, but those traits they carry might not have the same exact alleles. So let's just say we have a specific pair of chromosomes. One of them has an allele for brown hair, and one of them has an allele for blonde hair. They are homologous because they both have traits for hair color, but they're not identical because one had blonde and one had brown hair allele. Are sister chromatids always what? You have to speak louder, I can't hear you. Sister chromatids would be identical. Think about when we make sister chromatids, we're making a direct copy of the chromatid there. During crossing over might make those homologous chromosomes differ from each other, and it might make the sister chromatids not perfectly match anymore, but when we created those sister chromatids, they were identical. I see your point. All right, we'll make an exception then. Sister chromatids are not always identical because crossing over can change them. Okay, I'll go with that. I see your point. I suppose if I asked, are sister chromatids always identical in mitosis, we would say yes. But if we're in the context of meiosis, then no. Thanks for pointing that out. All right, 92. What is the purpose of the spindle fibers in mitosis and meiosis? We're going to use them to move the chromosomes. So, let's see if I have a picture somewhere. Whenever we see something like this, where we have these little lines pulling our chromosomes, those are going to be our spindle fibers. So whenever you see those lines pulling chromosomes, those are spindle fibers. So that tells us the purpose of spindle fibers is to move our chromosomes and chromatids. Did you guys talk about retroviruses? All right, well, I'll just tell you the answer and move on. Retroviruses would be use their RNA to make DNA. And you guys didn't talk about this either. It's the opposite of the question we asked earlier. If a host cell of a virus is not actively producing viruses, then it is in the lysogenic phase. But I don't think you guys need to know this, so don't worry about it. '95. Where is the Tata box located? Promoter. So C. Promoter region of the Tata box are going to be that area on the DNA where RNA polymerase knows to start making our mRNA transcript in transcription. So this is in transcription. '96. A silent mutation. Could it cause a change in an amino acid? Not A. Does it cause a premature stop codon? Nope. Could it cause a frame shift? Nope. So that means D. It produces the same amino acid. So if my DNA was this, and we had a substitution that changed it to this, and I don't know off the top of my head what amino acid this actually is coding for, but theoretically, let's say that these both coded for valine. If we had a substitution that made it code for the same amino acid, it would be silent because we wouldn't see the effect of it. What? Right, nothing changed. But we had a change in the DNA, but not a change in the amino acid. And I don't think this is actually the code for valine, I just, as an example, I don't have the chart memorized, so. '97. What is the most abundant type of RNA? I just gave away the answer. Is A, rRNA. I always remember this because rRNA makes up ribosomes, and we have lots and lots and lots of ribosomes in the cell. So that helps me remember that rRNA, or ribosomal RNA, is the most abundant. '98. Codons are. Are they on? What are they on? mRNA. All right. 99. Okay, we have a calico female cat and an orange male. And if we remember the problem we did earlier, we know that calico cats is X-linked. So if our female is calico, that means both of her X's are going to have a different allele. So one X is going to have the black allele, and one X is going to have the orange allele. And then we need to figure out the male's genotype. So we know he's going to be XY. And if he's orange, that means his X is going to carry the orange allele. So we want to find the possibility of having male black cats. So I'm going to set up my Punnett square. And I'm going to do the cross. And we want to find the possibility of male black cats. So this is another one of those problems where they're asking us for a specific gender. If we only want males in this case, we're going to exclude the females. So we're just going to look at the male genotypes. And we want to find out the possibility of those males being black. And we see that one of them, out of the two males, is a black cat. So we have a 50% chance of being a male black cat. All right, number 100. A man has B- blood type, and he marries a woman who is A positive. They tell us that they have a son who is O-. We want to figure out the genotypes of the parents, and then we want to find out the probability that they have an AB positive child. So first of all, we need to take the son's information to figure out the man and the woman's genotype. So first of all, we know that A and B are co-dominant over O, And we know that the positive Rh factor is dominant over the negative Rh factor. So because the son has both recessive alleles, we know his genotype is going to be OO and is going to have 2 minus or 2 negative Rh factors. So because of that, we could figure out the parents' genotypes pretty easily. So if the son is O, it's going to be like the problem we did earlier. We know that both a man and a woman would have to be carrying an O for them to have a son with the O blood type because O is recessive. Also, we can already figure out what both the man and the woman's second Rh factor is going to be. If the man has B minus, we know that he has to have both B minuses. But the only one we weren't 100% sure about was the woman because she could theoretically be heterozygous or homozygous for plus. But since they tell us that the son is O minus, that means she's going to have an allele for O minus also. So she's going to be heterozygous for the Rh factor. So now that we figured out their genotypes, we're going to have to treat this like a dihybrid cross because we're dealing with two traits. We have blood type and Rh factor. So we're going to have to figure out what possible combinations the man and the woman can make. So the only thing that, well, I guess the two things that man could make would be B minus or O minus. And we're doing this like we would do any other dihybrid cross. We could do it by foiling. The woman could make A plus, A minus, O plus, or O minus, because she's heterozygous for both. And if we wanted to verify that using the 2 to the n rule, we see that she has 2 heterozygous combinations. 2 to the 2 is 4, so that gave us our 4 combinations. The man was only heterozygous for 1 trait, so that's 2 to the 1, which is 2, which we verified he has 2 different possible combinations. Okay? So now we figured out what we're going to put in our Punnett square. So we have B minus O minus. And now we can do our cross. Our question is asking us, we want to find a chance we have an AB positive child. So if we're AB, we could have a couple different genotypes. We could be A positive, B positive, or we could be any combination of this. Because we know that the plus is dominant over the minus. So anywhere we see a plus and a minus, we know that they're going to display the plus. So if we're looking for somebody who is AB plus, we only have one here. This individual here would be AB positive because they have an A allele and a B allele, and they have at least one positive, which is going to be dominant. So that means we have a 1 out of 8 chance of being AB positive. Why we said he was O negative negative? Think of like if we're treating it as a dihybrid, we're gonna FOIL. Right. He has, both of his O's also have the negative. We know the son is going to be homozygous for O because he's O type and O is recessive. So in order to have O type blood, you'd have to have two O's. The same thing goes for a negative Rh factor. Negative is recessive, the plus is positive. And since we're diploid organisms, we always have two copies of an allele. So since he's O negative, we know he's going to have two alleles for the negative Rh factor. Does that make sense? She could make those four possible combinations in her gametes. She could pass on an A and a positive Rh factor to somebody. She could pass on an A and a negative Rh factor in her egg. She could also pass on an O and a positive or an O and a minus. Think of, it's kind of difficult to see that she's heterozygous for both because we're using all these weird symbols. It's not as easy to see as like a big A and a little a and a big B and a little b. But think of the fact that she is heterozygous for both, so she's going to make four different combinations.
So negative is recessive?
Yes, negative is recessive. It's not necessarily that she had an O negative, it's just she had both, and the negative could be passed on with the O or with the A. Does that make sense? So yeah, this is probably a more tricky dihybrid cross you're going to have because not only are we doing a dihybrid cross, which was kind of hard to begin with, but we're throwing blood type in there also. Okay, so that was the last question. We went through the whole test. Anybody has any questions? You can ask. Yes? As far as I know about the XP, it has something to do with we're unable to fix thymine dimers. Thymine dimer would be like a specific type of mutation, and we can't fix them when we have that disease. That's all I know about it. A thymine dimer is when two thymine nitrogenous bases are next to each other, and instead of bonding to the complementary strand, they bond to each other, and they create like a bump in the DNA strand. And that causes issues. And for whatever reason, when you have XP, you cannot fix those. Okay? Yes, I think so. I'm not 100% sure on details of Fragile X. So thanks everybody for coming. I have all the papers up here. If anybody wants to take pictures or if you miss anything, you can come get it. But everything's up here.
I want to take pictures, actually.
Go for it. Here, let me just spread them out here.
No probs.
Paul,
You're
about
taking
your
pictures?